Hello everyone, have a good day. My name is Seher and today I'm going to explain the production of an expression vector for the mammalian cell. Let's start the presentation. Starting over with the presentation, the topic of our presentation is production of mammalian GFP expression vector containing neomycin resistance gene. So, the first question is, what is vector? Vector is basically a vehicle that is used in various biological techniques to transfer a particular molecule of DNA into the host organism. What is gene? What is genes? So do you know gene is a hereditary material, right? And the genes which we will use are GFP gene and the neomycin resistance gene. GFP means green fluorescence protein gene, and the neomycin resistance is the gene which provides resistance against neomycin antibiotics. So next is the function of GFP gene. GFP gene is basically a gene which emits green fluorescence light when it is exposed to the light of certain wavelength. The main objective or the aim of this project is that we will insert the GFP gene as well as the antibiotic neomycin resistance gene into the host cell. While we will incorporate both of these genes, we will check the expression of the cell as well as the behavior of that mammalian cell in which these two genes are incorporated at the same time. Strategy. So, two type of vectors were used, PE-GFP and one vector which contained the GFP gene, and BCD and A3.1 vector which contains the neomycin resistance gene. These vectors were cutted with the help of restriction enzyme. So vector is also a piece of DNA, and the restriction enzymes are used to cut the DNA or the piece of DNA of a particular wavelength, and then they were run on the agarose gel to separate the pieces of DNA of different wavelength. So extraction fragments were taken from the agarose gel. The DNA which was to be cloned of that GFP gene and the neomycin resistance gene were also coated with the help of restriction digestion enzymes. Okay, and restriction digestion is a process basically which uses into various endonucleases enzyme to cut the pieces of DNA of different wavelength of different length. Products that were cutted were then extracted in 1% agarose gel using QIA quick gel extraction kit. Now, when we insert the vector and the gene of our interest in one place, we have to ligate them. For that purpose, typically 100 gram of vector DNA was used, and then where it was inserted 2 to 5 holes with 200 units of T4 DNA ligase in the total volume of 100 milliliter, and the temperature was 16 degree for 16 hours. 1 milliliter of ligation mixture was then used to transform almost top 10F competent cells. Bacterial transformation. 1 milliliter of DNA product from the ligation mixture was mixed with 100 milliliter of top 10F competent cells. These tubes were incubated in the incubator of ice for 30 minutes. Okay, and the timing was 60 to 90 seconds. After 2 minutes, we will add 950 milliliter of Luria Britannia, which is a medium, obviously, and it will basically provide a medium to the process of transformation. Transformation means the transfer of genetic material. Now these tubes were to be placed in the incubator at 37 degree for about 1 hour, and the spinning speed was 250 rpm. Spread aliquots of 100 milliliter of the culture on the LB plates. LB plates is basically the agarose medium in which we will spread those in which we will spread those aliquots, and it also contains the plates also contains antibiotic which is ampicillin, and then it is incubated at 37 degree overnight. So QC validation. What we will do in this process, We will inoculate 4.5 ml of LB solution which contain the ampicillin with a single colony, and we will allow the suspension to grow at 37 degrees centigrade for overnight. DNA was then prepared with the help of AcuPrep plasmid extraction kit. The method that was used was electroporation transfection method. In this method, electrical waves are used or electrical pulses are used to create the pores in the membrane of the cell, and that's how the DNA enters into the cell. Thank you. This is all from my side. Thank you so much.